In this third video of this week's topic, which is on gene expression in eukaryote, we're going to take a look at the post-transcriptional events. And as we know early on from the first video, there are 7 points of control. One of them is actually during post-transcriptional. So these processes could modify primary transcript prior to protein translation. They are capping, RNA splicing, which is of course polyadenylation, coupling of transcription and RNA processing within nucleus, not the coupling of transcription and translation in prokaryote, RNA transport, and of course, translation. So this is basically your sequence of DNA with its exon and intron, with some enhancers, some proximal control element, transcription side your promoter and whatnot. And of course, you have downstream, which also contain your polyA signal plus your termination region. So during transcription, what happens is that we have a pre-mRNA or primary RNA transcript, whereby it needs to be capped and basically to be added with polyA tail or polyadenylation. And then later on it will be processed to remove the introns over there, which is not needed. And then neighboring exons are joined together, which makes the coding sequence. And then you have it, your mRNA just ready for it to be expressed. So the first process that we're going to take a look at is actually capping. I think in another topic, 2-3 weeks from now, we are going to look at capping as well, again as a form of editing. So capping is actually comprised of 3 enzymatic reactions. It involves RNA triphosphate, which remove leading phosphate group from the mRNA 5' terminal triphosphate group. Second enzyme is the guanylyl transferase, which is the capping enzyme itself, which add the GMP to produce guanosine cap in a process called guanylation. And the third one is the guanine 7 methyltransferase, which methylate the guanosine cap so that it is attached to the 5' end of the transcript. So as we know, generally from previous video, that capping protects mRNA from the 5' exonuclease activity, which means that those that cuts from the outside, and it also prepares the transcript to interact with ELF2 to position mRNA on the ribosome. ELF2 is elongation factor. So now for the capping enzymes, basically they are the ones that gets the capping process going. So how it works, as you can basically view from here, as this is actually the explanation. So what happens is that gamma phosphate at the 5' end of the pre-mRNA is removed via phosphohydrolase. So the 5' link guanylate residue is added to the 5' end. Oops, this is where it is being added. So association of the capping enzymes with RNA polymerase through phosphorylated CTD will make sure that this is going to go through the second process, which is the splicing. So methyl group added to N7 of the 5' guanylyl and 2' hydroxyl groups of the half nucleotides is actually in the primary transcript. So the capping mechanism, again the diagram. So hopefully you could understand it much better. So capping starts from here. Similar process, winding, unwinding and capping. So this is done by methyl guanosine and also by the 3 enzymes in sequence order. Now we know that after capping genes are split. So what happens is that because there are lots of introns, and I think there is one slide showing of how big the intron is as compared to the exon. So intron, as the name says, is actually an intervening sequence. So it intervenes. It has lots of chunks of sequence, replicated sequence, which may be good, which a lot of time is not needed. So that's why it needs to be split out, or basically it needs to be removed. So as an example, actin gene has around 309 base pair intron that separates the first 3 amino acids and the other 350 or so, which means that it's just a matter of 3 amino acids in between them, but the size of the intron is actually very big. And an example, chicken pro-alpha-2 collagen gene is about 40 kbp long, with 51 exons of only 5 kbp in total. Now, the exons range basically varies in size from 45 to 249 bases and could be more than that. So the mechanism by which the introns are excised and exons are spliced together is complex and of course must be precise to ensure that the gene could be expressed later on and could work. Okay, so this is what I was talking about. So this is actually a comparison, of how the genes are split, between exon and intron. This is basically, I actually forgot, which genes this is, but this is from the same gene, but the position of the introns vary. So the coding region, the exons, is similar. Usually it is conserved, between these three. But can you see, the introns in between them? It's quite large, and to the extent that it's making the whole DNA sequence long, but probably kind of like junk, if not removed. So that's why it needs to be spliced off. So capped polynylated RNA in the form of RNP or ribonucleoprotein complex is the substrate for splicing. Now, the term splicing again means that it is excised or cut, and the exons are then sewn together to form each RNA, or not. The term is sewn, but it's actually ligated, simply said. So splicing occurs only in the nucleus. It cannot occur in the cytoplasm, so everything needs to be done before it is transferred to the cytoplasm for translation. So the 5' end of an intron, usually in high eukaryotes, is always GU, in the sequence, and the 3' end is always AG. And don't ask me why, but this is quite common. So all introns, have a branch site as well, around 18 to 40 nucleotides upstream from the 3' splice site. You can refer to your notes, it's actually in the next slide. So the branch site, is the one that is essential for splicing, to form the lariat branch, and then it will be cut off. So this is what, I was talking about just now. So at the branch site, there is actually a very specific sequence, which is the YNY-RAY, and it is always the sequence that will be recognized, to be spliced out. So it will form a lariat branch. So this form is called lariat, and then it will be cut off at the hydroxyl site, and then you have your exons there, and then it will be sewn back together. So now, like I said, the branch site is always YNY-RAY, where the Y is the pyrimidine, R is the purine, and N could be anything. So it could be a pyrimidine or it could be a purine. So AG, you see. So the lariat, or the branching that you saw, is a covalent closed-up loop of the RNA, which is formed by attachment of the 5' phosphate of the introns invariant 5G to the 2' hydroxyl, OH, at the branch A site. So when the exons are then joined, excising the lariat. So the lariat is usually unstable after it has been cut off, hence, the 2' 5' phosphodiester is usually quickly cleaved, and the intron is degraded in the nucleus. So nothing will be left behind. So this is the molecular structure of the branch site and the lariat. Now, why is it important, the SNRNP in splicing? So because small nuclear ribonucleoprotein particles, or SNRPs, are the ones, that needs, or needed for splicing. So it is actually very small, consists of small RNA of around 100 to 200 bases long, and it has about 10 different proteins between them. So some of the 10 proteins are general, some are specific. So SNRPs and the pre-mRNA form the spliceosome, the machinery to cut off the exon from the two neighboring exons. So spliceosome is actually the size of ribosomes, and its assembly requires ATP or energy, adenosine triphosphate. So as you can see, so this is the spliceosome, and how basically it is being used to cut off the intron. So assembly of the spliceosome. So I'm just going to go into very general of the assembly. So this is the schematic diagram. The explanation is here. So the SNP U1 and U5, as you can see, so it actually starts from here. U1, and U5, U1, U5, bind at the 5' and the 3' splice sites. So one will bind here, one will bind towards the end. And U2, over here, binds at the branch site, the ones to be cut off. So interaction between the SNRPs, bring the 5' and the 3' splice together. So because the, I'm not sure what it's not, So your U1 is over here, your U2 is here, and your U5, is actually situated there. Okay, so what happens is that it basically will form a loop, okay, or the lariat, okay, and basically if you look at this again, it's actually going to be a bit difficult, okay, because they are actually showing how it has formed a loop by then, okay, but it actually your U5 is here, so when it forms a loop, then it will come there, giving this kind of impression. Now, so what happens is that, okay, when they are spliced, okay, so lariat can form and the exon's ligation could occur. So trans-esterification reactions, okay, is the one that joins the exons, okay, and it is of course, okay, catalyzed by the ribosome. So if you were to see along the way, and like I said, I'm not going to go into details into this, or probably there will be another form of spliceosome in gene editing, I think so. Okay? We will discuss that in details. But okay, in the machinery or in the assembly of the spliceosomes, okay, there are lots of SNPs needed. Okay, so we have U1 up until U6. Did you notice anything missing? Yes? No? So we have U1, U2, U3, U4, U5, U6. So my question is, where is U3? Find it out and probably we'll discuss it. Now let's move on to the last part, which is actually on the 3' polyadenylation. Well, polyadenylation always occur at the 3' site anyway, it couldn't occur at the 5', so it signals for termination of transcription. Okay, and this could only occur after RNA polymerase has transcribed past the consensus, okay, which is AAU AAA sequence, which is the poly A addition site. So around 10 to 30 nucleotides pass this site, okay, a string of around 100 to 200, basically the length varies, okay, adenine residues, only just A, okay, 100 to 200 A's, okay, are added to the mRNA transcript, okay, and this is your poly A tail, so that's why it's called poly A, okay, everything is just a string of A's. So poly A polymerase adds these A residues, and the function, not known actually for sure, okay, but again, like I said in the earlier videos, okay, this poly A tail may govern stability of the mRNA, and it has so far been proven, but the actual function of poly A tail is not really known, because usually if there is no poly A tail, it's still stable regardless, okay, but stability is actually much more enhanced with the addition of poly A tail. Now, the polyadenylation step, and again, okay, we will discuss this in detail as well, okay, in one of the gene editing topic. The mechanism, okay, is of course governed by a series of enzymes and factors as well. So we have transcription factors over here, similarly. Okay, so this is diagram, okay, for you, okay, to have an overview, okay, and the explanation is actually over here. So we need the cleavage and polyadenylation specificity vector to bind upstream to the AAU AAA site, okay, at the 5'. So then cleavage stimulatory factor F, okay, will interact with downstream GU, okay, so remember we have GU, okay, at the 5', okay, so, and bound to the CPSF to form a loop in RNA. So when it basically binds to another, it will form a loop. So cleavage factor 1 and cleavage factor 2 will work together with CPSF, and of course, okay, this will slow polyadenylation process. Why we need to slow it down? Okay, because, okay, poly A polymerase, okay, will have to stimulate cleavage of the poly A sites, okay, which means that, okay, it has to add in the strings of A, and then, okay, later on cleavage. So the polyadenylation process has to be stopped, because if it goes very fast, then probably it will not be completed. So poly A binding protein 2 will accelerate addition of A's by the PEP, okay, poly A polymerase, okay, and signal PEP to stop polymerization after addition of 200 to 250, okay. So usually it's around those kind of ranges. So like I said, okay, sometimes the size is 100 to 200, okay, but okay, the most that they have encountered is actually around 270 amino acid residues, A residues. So the poly A tails, okay, again, it controls mRNA stability and influences translation. Now this is basically just an overview, okay, of the polyadenylation diagram. So basically we have, okay, from the start, okay, whereby RNA polymerase binds, okay, and it is being transcribed, so we have a cap being there, and then, okay, we have this signal, okay, AAU AAA, and then you know that it signals for poly A to be added, okay, addition of poly A by the poly A polymerase or PEP. So after it basically has added between a string of 100 to 200 up until 250 amino acid residues, so it will be cleaved off. Right, so this diagram represents the whole process, okay, the three processes, okay, from capping, splicing, to cleavage and polyannulation, plus, okay, A to I editing. So I'm not going to go into details, okay, for this yet, okay, so but don't just forget it and don't just, okay, I don't need to remember that, but we will discuss this in detail, especially when we go to RNA editing in one of the topics in about 2-3 weeks time. Okay, so this is the end of the third video. Move on to the fourth video.